All right, so welcome to our second lesson. We're going to talk about something called signal transduction, which is basically carrying a message inside of a cell. So let's get to it. What is signal transduction? It's the mechanism by which you have this chemical signal that a cell receives, which is then going to lead to a specific response. So it's basically the integration that's happening inside of a cell. And it's usually a bunch of molecules interacting with other molecules in very specific ways. And before you go, oh, that's specific, we're going to go into that in a little more detail. So signaling molecules and the receptors for them are pretty heavily conserved across a lot of species. Bacteria have signaling molecules and receptors that are very similar to our own in some cases. So this probably arose very early in evolution and was probably useful for unicellular creatures to communicate with each other. Then once multicellular creatures arose after the evolution of eukaryotes, then these signaling pathways were then co-opted to have cells be able to interact with each other. So that way you could have all these multiple cells, millions of cells, working together to create a functioning organism. All right, so let's start talking about some of these details. So, signal transduction happens in a series of steps, very similar to what I already talked about. It is reception, which is basically what I was talking about, the stimulus. I was just trying to be more general. Transduction, which is when I was talking about integration. And the easiest one of all, response, which is the same. So this is where you get a signal or a message from the surface of the cell to the interior of the cell so the cell can actually perform some function. So we're going to look at this quite a few times. So we have the external surface of a cell here. A signaling molecule comes, binds to a receptor. Remember, the other word for a signaling molecule is going to be ligand. So this is a ligand that matches with that receptor. When it binds to the receptor, you're going to have reception, getting a message. The next step is transduction, where that receptor is going to then change a molecule, which is going to change another molecule, which is going to change another molecule, and it's basically like passing a message back and forth. So if you've ever played the game telephone, where you and a bunch of people just kind of stand or sit in a line and you have to pass a message from one person to the other, that's kind of like signal transduction, except it's a lot more accurate than telephone, because you're not like trying to say words. And then finally, you have whatever response that cell is designated to do. We're not going to get super specific about responses just because depending on the cell, depending on the signal, you have different responses. So let's talk first about reception. So a receptor and a ligand fit together, causes a change in the conformation of the receptor, and the receptor will then cause a change in the conformation or shape of some other molecule inside of the cell. But first, we've got to talk about these receptors. There are three major classes of receptors that respond to ligands. There are G protein coupled receptors, sometimes called GPCRs. It's just basically an acronym. There are receptor tyrosine kinases, sometimes called tyrosine kinase receptors. So I think they are often abbreviated TRKs. And then there are ion channel receptors, which don't have their own specific abbreviation. And it depends on what molecule they're transporting. What ion they're transporting. So let's take a look first at G protein coupled receptors. We have thousands of these in our body. So we have thousands of our genes are devoted to making these guys. They're very important. They probably evolved very early in cell signaling because every living creature on the planet has some sort of G protein coupled receptor. They're they go through the entirety of the cell membrane, but they're not a channel. They don't allow something to pass through the cell. So they're going to have a ligand bind on the extracellular or outside cell surface, and then some of them will have molecules that will bind on the internal or cytoplasmic surface. Some of these receptors are like epinephrine receptors. Epinephrine is noradrenaline, I believe. Hormones, neurotransmitters, a bunch of these are going to be GPCRs. So there's going to be something, since they're coupled to it, there's going to be something called a G protein that we'll talk about in a few minutes. So what ends up happening is a G protein coupled receptor has nothing bound to it, but then a ligand comes. That signaling molecule comes, and it is going to activate that receptor. That receptor is going to pick up a neighboring molecule that's just hanging out in the membrane called a G protein. It's called a G protein because it has either GDP or GTP attached to it. A GTP is guanosine triphosphate, very similar molecule to ATP. It can actually be used to transfer energy, and in this case it's used as an on-off switch for a G protein. So a G protein, when it has a GDP bound, remember GDP has only two phosphates, it's going to have lower energy, just like ADP has lower energy than ATP. When you have a GDP bound, that's less energy, you can think of that as like the off switch. So if a G protein has GDP bound to it, it is not active. It's not doing anything. On the other hand, when a G protein coupled receptor is activated, that allows it to bind to this inactive G protein, and that causes it to change shape so that it kicks out the GDP and then binds a GTP instead. Remember, GTP is going to have more energy in it, just like ATP has more energy in it. So you can think of that as turning on the G protein. Once the G protein is turned on, it is still stuck to the membrane, so it's not going to go anywhere inside of the cell, but it can then move on to activate other molecules like enzymes. We'll talk about several different types, and then that will trigger some sort of cell response. Once that G protein activates an enzyme, it hydrolyzes the GTP to form that inorganic phosphate that you see here, which is, helps if I actually use the right side of the pen, and into a GDP. The GDP stays stuck to the G protein. Now it's got a GDP attached to it, means it's off now. In the meantime, while we were looking at what was happening with the G protein, the signaling molecule is not going to stay permanently attached to the receptor. It's not covalently bound, it's just loosely associated. So they're held together very loosely, so that way that signaling molecule can come off. When that signaling molecule comes off, you don't have that G protein coupled receptor active, it's not activating any G proteins, so in order to have the cellular response triggered again, you need more of the signal. So it's very important that you have the signal come in, you have a response to it, but that response doesn't stay turned on permanently. So let's look at this in action. So we have a G protein coupled receptor, it's inactive. We have a receptor, that's the blue. We have some sort of enzyme, that's the purple. And then we have a red signaling molecule. So that signaling molecule comes in and binds. When it binds, it allows that G protein to bind to it, trade out its GDP for a GTP, and become activated. And that's represented here by the color change. It went from that boring kind of dull green to a lighter green color, indicates it's active. Now, it's got a GTP attached to it, so that G protein is now going to release from the receptor. The ligand can also release as well. I did that too fast. Okay. Then the active G protein is released from the receptor. It can then go on to activate an enzyme nearby, which is going to trigger some sort of cellular response. So that's Notice we have our inactive enzyme, it's a darker purple. Our active enzyme, on the other hand, is that lighter purple, just to kind of show you what's happening. Now, we have that cellular response going on. In the meantime, the G protein, once it's activated, this other enzyme is going to hydrolyze its ATP. So it dissociates from the enzyme becomes inactive, it's going to break down its GTP to make GDP, which inactivates the G protein until another signaling molecule comes along. Oh, sorry, I guess they're RTKs. I've seen them abbreviated as TRKs as well. So receptor tyrosine kinases are actually something that is not found in all living organisms. They're only found in eukaryotes, so kind of an advancement that happened when eukaryotes evolved. So bacteria don't have these. What happens is these exist as single receptors hanging out in the membrane, but when ligands bind, you actually need 2 receptors and 2 ligands. When both receptors have ligands bound to them, they actually attach to each other and they form a dimer. Remember, di means 2, like carbon dioxide. So it's a dimer, meaning it's made of 2 receptors stuck to each other. Those receptors then phosphorylate each other, by which I mean that they add phosphate groups to a specific amino acid. They add phosphate groups to tyrosines that are on the other receptor, hence the tyrosine kinase. Those tyrosines that are phosphorylated can now activate a bunch of different relay proteins, and each of those relay proteins could trigger a different response in a cell. Just kind of depends on which ones are found in that cell. Receptor tyrosine kinases are often going to be a malfunctioning gene or a malfunctioning protein in a lot of different types of cancers. So here is our overview. We have in panel 1, we have a ligand coming in to bind. You have these inactive individual receptor monomers. Notice they have these tyrosine bulbs. Those are going to become important when a ligand binds. So ligands bind to 2 different tyrosine receptor kinases. They form a dimer, and then when they form a dimer, that activates the receptor, which then phosphorylates itself, or they phosphorylate each other. So now you have these phosphorylated tyrosines, which are an active protein, which can then phosphorylate other molecules, and these molecules can then go initiate other cellular responses. So let's look at this in action. So you have 2 receptors, you have 2 ligands. When the ligands bind to the receptors, those monomers then dimerize, and those tyrosines become phosphorylated. So, you saw 3 ATPs come in. I decided to save time and not put the other 3 coming in. So now what ends up happening is you have these relay proteins. They come, they get activated by these phosphorylated tyrosines, and then they're able to go off and they're able to perform some sort of cellular response. Looks very complicated, but when you start digging into it, it's not as complicated as it looks. And then ion channels. So what happens is with an ion channel, they're basically gated doorways into a cell. They open in response to a specific signaling molecule or ligand, just like you can open a specific door with a key. When they open, they will allow only a specific ion to pass through. So you can have ligand-gated potassium channels, sodium channels, chloride channels, calcium channels. It just depends on which cell type you're looking at. So once a ligand binds, opens up a gate, that's going to allow ions to pass through. That can change concentrations of ions, that can change electrical charges, that can allow things like calcium to come in and affect things further on in the cell. So there's a lot of things that can happen when these ions move in or out. And so then you have the ligand dissociate, and then the gate closes up. So let's look at that in action. So we have our blue receptor, notice it's closed, nothing can pass through, and we have a bunch of red ions, a high concentration outside of the cell, a low concentration inside of the cell. When the ligand binds, it opens up the ion channel, which then allows those ions to diffuse down their concentration gradient. That binding of the ligand is not permanent, so it can dissociate. And when it does that, the channel is going to close, so now ions can no longer pass through. If you want to open that ion again, you're going to have more ligand come by and associate with the receptor. So we talked about the reception. Now we need to talk about transduction. There are these molecules called second messengers, which are all inside of the cell that carry the signal further on into the cell. The first messenger is the ligand and the receptor. Those combined make the first messenger, and then they pass on their message to the next messenger in the cell, which is the second messenger. There are a number of these second messengers that are commonly used in a lot of cells, and we're going to look at them. So they're not proteins, they're water soluble, but they can't pass the membrane, and they can diffuse through a cell to pass on a signal. There are three major ones we're going to take a look at. There's cyclic AMP or CAMP. There are calcium ions, and then there's inositol triphosphate or IP3. So cyclic AMP, the AMP should hopefully be a little familiar. That's adenosine monophosphate. So it's an adenine, a ribose, and a single phosphate attached. Then you have a specific enzyme called adenylyl cyclase, which is this happy little fella up here. Where is my cursor? There we go. So adenylyl cyclase takes an ATP. It's going to hack off two phosphates, and then it's going to make cyclic AMP, which is kind of circular. So cyclic AMP, the cycle part, is this part right here. It's not a giant circle, but it's still different in structure. That cyclic AMP usually goes on to activate a specific protein kinase. A kinase is going to be a protein that adds phosphates onto another protein. A phosphatase is going to take those off. So a lot of G proteins are going to activate adenylyl cyclase, and when they do that, they're going to cause the formation of cyclic AMP, which then causes the activation of protein kinase A, which then goes on to do a whole bunch of other stuff. You shouldn't have to have memorized that cyclic AMP activates protein kinase A. You do need to remember the fact that adenylyl cyclase makes cyclic AMP. Calcium ions are another second messenger. They can also be released by the action of a second messenger, so calcium ions can play a number of roles. So before we look at this, let's look at how calcium concentrations are inside and outside of a typical cell. Typical cells are going to contain low concentrations of calcium inside, but they may have organelles that have higher concentrations of calcium. So one very specific example is the endoplasmic reticulum. You can have specific sections of the endoplasmic reticulum whose function is to store calcium. So cells will actively pump calcium outside of the cell to the extracellular fluid, which will then have a high concentration, or they will actively pump calcium into specific organelles, like, for example, the mitochondrion here, or into like specialized segments of the ER. When calcium is released into the cytoplasm of a cell, it's going to trigger some reactions that basically, this pathway involves one of the other second messengers, IP3. There's another second messenger called diacylglycerol, or DAG, but we're not going to really mention it very much. And then these pumps are going to act to reset concentrations of calcium again. So here's what happens. I'm sorry, the animations aren't as fancy as the other ones. First step is a signal molecule binds to this G protein coupled receptor. That G protein, when it's activated, is then going to activate a molecule called phospholipase C. You don't have to have memorized what that is. what you need to know is that it creates, basically, it's going to create this IP3. IP3 is a second messenger that will then go, DHE goes off elsewhere. We're not going to pay attention to it now. IP3 diffuses through the cytosol, and it's going to open these gated calcium channels inside the ER membrane. When those open, that's going to allow calcium to flow into the cytosol, and that calcium can then activate other proteins. So you'll see we have both a G protein coupled receptor in this pathway, and we have a ligand-gated ion channel as well. Only thing we're missing is a receptor tyrosine kinase. I know this all looks convoluted, this all looks really complicated, but it's the series of reactions and stuff that help your cells function. It is pretty complex. There are scientists who devote their entire careers to trying to isolate these individual pathways. And then before you're like, oh my God, they're wasting their lives. But then these pathways are very important because when there are malfunctions in them, a lot of times you have diseases or you have cancers or things like that. So knowing how these pathways work allow us to develop effective treatments or preventative measures for diseases. Then finally, you have a response. There are a number of things that can happen. You can activate genes. That's usually done with these molecules called transcription factors. You can cause physical shape changes in the cell, or the most dramatic type of response, you can trigger apoptosis. I'll tell you why that's spooky in a minute. So, these are some of the different types of responses. I already kind of went over those. Only thing that's different is we have our definition of apoptosis here. It's programmed cell death. So there are times in a cell's existence where it does need to die. One important example is in development. We'll look at an example of that here. But then if a cell has catastrophic errors in its DNA and it is likely to become cancerous, there should be signals, there should be pathways inside the cell that allow it to program its own death so that way the rest of the organism can survive. So here we see an interesting example of signal transduction in yeast. So yeast actually have, they're not genders per se, they're two different mating types. And yeast have to interact with yeast of the opposite mating type in order to mate. And yes, I know it's weird because they're single-celled and yet somehow they're mating. I was weirded out by it too. But anyway, what happens when they have the appropriate mating factor, it actually triggers this complicated pathway that you see here. It's a G protein coupled receptor, which then activates a G protein, which does some phosphorylating, which phosphorylates some other things. And that phosphorylation ultimately leads to the building of microfilaments. Microfilaments then change the shape of the cell and form these kind of weird bulby structures that are called shmoos. That's the most fun word that I've seen in biology in a while, shmoo. So this is a cellular response. You have a signal come in, which is the proper mating factor, and it triggers a response. The cell changes shape. Phosphorylation is just adding a phosphate. So here we have a kinase adding a phosphate that can activate a protein. So if a protein is off when it's not phosphorylating it can turn it on. If you can also have phosphorylation that triggers inactivation. Just depends on the protein you're looking at, though in general, most of the time when you add a phosphate, that turns on a protein. Phosphatases are going to take off phosphates. So all of these reactions that you've seen are reversible. A ligand can bind to a receptor and then it can pop off. A G protein can become activated and then it can be inactivated. Adenylyl cyclase can be activated and then be inactivated. Protein kinases can be turned on and off. Phosphorylation can be reversed. So none of these are forming any permanent changes. All right. So we're going to look at a bit of a phosphorylation cascade, which is a series of molecules phosphorylating each other. So we have a signaling molecule that's got a receptor bound to it, and then that activates a relay molecule, which is going to then phosphorylate or activate a protein kinase. That protein kinase, when it becomes activated, can then activate the second protein kinase. That second protein kinase, when it becomes activated, can then activate a third protein kinase, and that third protein kinase can then activate some other protein, which can then trigger a cellular response. So a phosphorylation cascade is basically a bunch of things phosphorylating each other, and we'll talk about why that's important in a minute. So here we see an example of a phosphorylation cascade. The very first signal transduction pathway that was ever isolated was basically the binding of epinephrine to its receptor. So it will be a very common one to see in college if you're looking at like biochemistry, or if you're weird like me and you end up taking a signal transduction class. Anyway, so you'll see you have these phosphorylation cascades that happen. You've got different molecules being phosphorylated, like, for example, you have protein kinase A here. You've got a phosphorylase kinase, and you don't have to know what that is. I don't know what it is. You've got this glycogen phosphorylase, and then what that ends up doing is that ends up producing a bunch of glucose molecules. So epinephrine, when it is released, triggers your liver to start releasing glucose from glycogen, and this is how that works. So usually one of the potential ways that you can trigger a difference in a cell is that you can have transcription factors activated. A transcription factor will determine whether a gene is like activated and is making a protein, or whether that gene is not being read and not creating an active protein. So a lot of these phosphorylation cascades, their ultimate target is going to be a transcription factor. They're going to activate it, cause a protein to be produced. Apoptosis is a very important process. It is natural for cells to apoptose. There are, in fact, diseases where we're speculating that it's a failure of cells to apoptose properly that causes these diseases. And then if apoptosis is not activated when needed, you can have tumor cells or cancer cells that can then divide out of control. Interesting thing, when you start, there's actually, I think it's called the death receptor. I don't know if that's its official name, but it's kind of ominous sounding. But when it's activated, that starts triggering, hey, we're going to start the apoptosis process. So that will start to basically activate these proteases. Proteases break down proteins. Nucleases are activated too. They break down nucleic acids. And then basically the cell starts to form these blebs. I know there's a lot of weird words today, which are these little bulgy things sticking off, and the cell will potentially fragment, break into little bits that can then be cleaned up by the immune system. And then finally, you also need to turn off signaling. So like I said, everything in these pathways can be reset. So ligands can unbind from receptors, G proteins become inactivated and bind GDP. Tyrosine kinases can then be dephosphorylated so they can be activated again. Ion channels usually are only open for a specific period of time. There are phosphatases running around dephosphorylating proteins, and all of this basically resets the pathway so you can get another signal coming in. So, why all this complication? Well, one reason why you have all of these different molecules in these like phosphorylation cascades is it actually allows you to amplify the response. So here's our example here. We have a single molecule of epinephrine come in and bind to a single receptor. That activates a G protein coupled receptor. That receptor can then activate 100 G proteins. So each of those 100 G proteins can then go on to activate 100 adenylyl cyclases. But each of those adenylyl cyclases, while they're active, can make multiple cyclic AMPs. So at this point, we are now making like 10,000 cyclic AMPs. Those 10,000 cyclic AMPs can then activate 10,000 protein kinase A's. Those protein kinase A's will then phosphorylate 100,000 other molecules, which will then phosphorylate 1 million of these glycogen phosphorylases, which will then produce, I think it's 100 million molecules of glucose. All of that from one individual signaling molecule. So it allows you to amplify or ramp up your response to a very small signal. Also, you can have multiple responses that come in from the same signal. So depending on what signaling molecules you have, what second messengers you have, what receptors you have in a cell, each cell can respond to the same signal differently. There are multiple pathways. These pathways can interact. And so you can have like 2 signals come into the same cell, like what you see here. 2 signals coming in, they can interact with each other so that they produce a different response. You can have, for example, this is all with the same signaling molecule in each of these examples. You have a signaling molecule that binds to a receptor, activates certain relay molecules, and creates a response. In a different cell, you can have same receptor, same signaling molecule, but they activate a different second messenger, which then activates other proteins, which produces new responses. We already looked at this third example with cell C. You have 2 different pathways, 2 different ligands, 2 sets of receptors, which can then produce a different response. And then you can even have another receptor that's completely different, that responds to the same ligand and activates a completely different set of responses. So you get a lot of versatility with this. You can get a lot of different reactions in different cells just by releasing one molecule into the bloodstream. In order to make this more efficient, you can have signaling complexes or scaffolding proteins, which basically just keep all of these kinases that need to activate each other in the phosphorylation cascade, that keep them all in the right order. If you remember when we were watching those videos in class of what the inside of a cell actually looks like, it's very crowded, and if you rely on stuff to diffuse through it, it's going to take a while. So instead, if you keep everything that needs to interact in the same part of the cell, you can make this reaction happen faster. That is it. I would watch this more than once because I threw a lot of information at y'all.